Hello and welcome everyone. I am Vinay Kumar Singh and I am going to present a PPT on plant DNA extraction to PCR amplification, because we need to identify a plant, or in other cases we need to identify bacteria or fungus. So DNA extraction is very important, and in PCR amplification is very important, because after PCR amplification and sequencing you get the sequences those you used to blast to identify or constructing trees. Okay, so here is the first thing you need to do is plant DNA stock chemicals. Okay, firstly you will have to prepare the plant DNA extraction stock chemicals. First of all, you should prepare stock solutions, because it is not that every time you are going to measure the large amount of chemicals, or you will be adjusting pH every time. So stock solutions are very helpful. You just make your stock solutions, and every time you are going to extract DNA, you just dilute these your stock solutions and use freshly. Okay, so there are 4 stock solutions in plant DNA extraction. The first one is 1 molar tris HCl. For a preparation of this 1 molar tris HCl, you need to if you are going to make the final volume of 50 ml, then take 6.05 gram of tris HCl and add it into the 40 ml of double distilled water, and using the NaOH adjust the pH to 8 and maintain the final volume to 50 ml. Okay, so your 1 molar tris Cl will be finally 50 ml and of pH of 8. Okay, and the second important chemical is 0.5 molar EDTA. For preparation of 25 ml of 0.5 molar EDTA, you will need 4.65 gram of EDTA, and like the previous one, you will need to add it into the 40 ml of double distilled water, and you will be adjusting the pH to 8 by using NaOH. Okay, and the final volume will be 25 ml. Then the third stock is 5 molar NaCl. NaCl is also an important chemical, and you will have to prepare a stock, because usually the box you get is 1 molar, so you will have to prepare a 5 molar solution. For 50 ml you will need 14.6 gram of NaCl, and you will be adding this amount in 50 ml of double distilled water slowly. So you need precautions, because NaCl when you will adding this into the double distilled water, the your solution will be heated. So I will suggest to use the plastic vials, okay, not that of glasses. The fourth one is 10% CTAB. For preparation of 10% CTAB, you need about 5 gram of CTAB, and you will be adding it into the 50 ml of double distilled water, and your 4 stock chemicals will be prepared. The second one is CTAB DNA extraction buffer. We first prepare the stock solution, and from these stock solutions we will be preparing the CTAB DNA extraction buffer. The chemical you need to prepare CTAB DNA extraction buffer is 1 molar tris Cl, 0.5 molar EDTA, 5 molar NaCl, and 10% CTAB, finally beta mercaptoethanol. Okay, so here I have prepared a list of the amount of chemical you will need to make 25 ml. If you want to prepare CTAB DNA extraction buffer 25 ml, then add this amount of these chemicals in this ratio, and for 50 ml you just double the amount, and for 100 ml you just double the amount of 50 ml. Okay, so if I say from my practical point of view, then you will need about you can do actually 40 to 50 sample you can use by preparing a 25 ml of DNA extraction buffer. Okay, so you can extract the DNA from 40 to 50 samples using this 25 ml. Accordingly, you can design your amount of extraction buffer you need during your DNA extraction protocol. The third one is how you are going to use the CTAB DNA extraction buffer to extract the DNA. These are the steps. The first one is you will be crushing about 100 milligram leaf tissue. I will suggest that don't take very little amount in DNA extraction, otherwise you will be finally getting the very low amount of DNA. And do not take very large amount of leaf samples or anything. Any sample if you are doing with bacteria or fungi, because larger amount will take great amount of your this extraction buffer, and this will also you will not actually using those DNA that you will found finally, because what our goal is to do sequencing, and you can do sequencing after getting 400 or 500 nanogram DNA, and with this 10 to 100 milligram of sample you can easily get that amount of your DNA, which you will need to do PCR. So crush about 100 milligram of leaf tissue and CTAB extraction buffer, and you will be taking this about 800 microliter plus PVP. PVP is used if you are getting your protein contamination. This you will realize after extraction of the DNA. And if you in the final step, when you do the NANODROP, you will find that if your value is somewhere is less than 1.8, that means your DNA is contaminated with proteins. Then for this purpose you will need PVP 2%, 2 microliter. You will mix this during your DNA extraction in that DNA extraction buffer. And side by side you need RNase A. This is also you will be using a 2 microliter, and this we need when your OD value will be larger than 1.8. then that means you have RNA contamination in your DNA sample. So if you are getting a protein contamination, then use PVP. If you are getting RNA contamination, then use RNase A. And if you are not getting protein, then you don't need to add PVP or RNase if you are not getting RNA contamination. So further, if you you crush the sample of 100 milligram in the extraction buffer and added PVP and RNase, and then you need to incubate this your sample at 65 degrees centigrade for 30 minutes in water bath, or you can also incubate it for 1 hour. The second step will be after 1 hour you take 8 microliter of phenol chloroform solution, 1:1 solution. After adding this, you will be doing centrifugation at 12,000 rpm for 20 minutes at 4 degrees centigrade, or you can also do it at your room temperature. After centrifugation, you just very carefully take the 500 microliter of supernatant without disturbing the middle phase, because middle phase and the lower phase will have protein or RNA contaminations, or they are the layer which having the organic phase. Okay, so you need not to touch that. Only take 500 microliter of supernatant in a fresh centrifuge tube. In that 500 microliter supernatant, add 500 microliter of CTAB buffer, which will also have 2 microliter of PVP and RNase accordingly if you are getting more contamination, and also incubate it for 30 minutes at 65 degrees centigrade in water bath. Okay, after taking it from water bath, then again add 800 microliter of CI, chloroform isoamyl, in the ratio of 24:1, and then centrifuge it at 12,000 rpm for 20 minutes at 4 degree or at room temperature. These centrifugation process, you can just change the time. If you if you are doing for a shorter duration, centrifuge at a higher speed, or if you are your RPM is low, then go for the 20 or 25 minutes or somewhere. After adding this CI and after your centrifugation, take 500 microliter of supernatant very carefully again without distributing the middle phase in 1.5 ml of label centrifuge tube. Add 800 microliter of chilled isopropanol, okay, and then 10 microliter of 3 molar sodium acetate, and mix gently and incubate it in -20°C for at least 24 hours or at 48 hours. Next day, after 48 hours, DNA strand will be visible in tubes, okay, or otherwise it will be invisible, or like mucilage. You can check at the bottom of that centrifuge tube. Centrifuge it at 10,000 rpm for 5 minutes. Very carefully discard the supernatant, because it is not that every time you get the pellet, DNA is invisible most of the time. So you will be at the time of discardation, you will be also discarding your DNA. So carefully discard the isopropanol, and then it add 200 microliter of 70% chilled ethanol to the pellet and keep it for 20 minutes at -20°C. Discard the ethanol carefully again and add 200 microliter chilled ethanol again to the pellet and keep it for 20 minutes at -20°C. Discard ethanol, or at this step you can also do centrifugation before discarding the ethanol. After centrifugation you can discard your ethanol. Discard ethanol, air dry the pellet for evaporating the ethanol. Generally just put it for 20 or 30 minutes, and after your pellet is dry, add 200 microliter of tea buffer, or just suspend it into the milky water, okay, and keep it in -20°C for further use, okay, when you want to do your PCR. Further, after your DNA extraction, you can also go for your quality check on the Agarose gel electrophoresis. If you are lucky, you will get the bands on your gel electrophoresis, and it means that your DNA strands are larger enough. Okay, because many times we get fragmented DNA, and that is not going to be good for your PCR. You will not getting band, or you are not going to amplify using the primer pairs. So quality of DNA is very important, and you can check it after your DNA extraction on Agarose gel. Sometimes you don't get the bands on your gel, and after that when you go for your PCR amplification, you got the result. So it is not like that your target gene is degraded, but quality check is important, and it is assurance that your extracted DNA is good, okay, it has larger fragments. Then number 5 is quantity check. Here the Agarose electrophoresis is for quality check, and NANODROP is for quantity check. You can by using NANODROP you can assess your nucleic acid concentration, purity ratios, like in 1 microliter what is the amount of your nucleic acid, like somewhere our friends are doing DNA extraction of fungus, and we see that they have 3 to 6 nanogram DNA per microliter. In plants we generally get about 300 or 400 nanogram per microliter, or sometimes we also get more than 2000 nanogram per microliter. Purity ratios are 260 and 280 ratios should be in between 1.8 to 2. This shows that your DNA is pure. If you are getting less, this value is less, then that means you are getting a protein contamination. For that purpose you will be using PVP, and if you are getting value more than 2, then that means your DNA extraction also have RNA contamination, and if your 262 to 30 value is more than 2, that means the phenols etc., that contamination is not there. Okay, generally we see that the ratio 260 and 280 is very important for PCR amplification, and this 260 and 230 ratio is generally not cause hindrance in your PCR amplification. So always look for the ratio of 260 to 280. So the sixth one is PCR amplification. After extracting the DNA, what we want to do is we amplify a region of our desired gene, either nuclear, either mitochondrial, either plastic. Like very popular gene we amplify for identifying species or making trees, ITS, which is a nuclear gene, and for plants we use RBCL and Maturase K. These are the plastid genes, and we know that fungus don't have plastids, so you are not going to find these genes in fungus. And for animals there is gene cytochrome oxidase first, which is generally used for identifying the animals. Okay, so for PCR amplification, firstly we need to prepare our reaction mixture. Generally people do in 50 microliter, but you can also get your results in 25 microliter. So in papers you will also get that the PCR reaction mixture of 12.5 microliter. So it is up to you what amount of PCR amplified product, in which amount you want to get. Okay, if you have enough amount of your extracted DNA, and you can use this for further, and your PCR amplification is very good, annealing temperature etc., means you are not getting failures, so you can just go for your 25 microliter. Okay, if you have a very rare sample, or you don't have you don't get the largest quantities of your extracted DNA, go for 50 microliter. If your PCR is success, you will have the large amount of your PCR product to do sequences for your sequencing amplified product. Okay, so for PCR reaction mixture there are 8 components you need. The first one is 10x buffer. This 10x buffer you generally get mixed with 20 millimolar MgCl2. You need it in amount of 5 microliter, then further 2.5 millimolar of DNTP, 2.5 microliter, then both forward and reverse primer of 10 micromolar in 1.25 micromolar. And fifth one is Taq polymerase. Generally it comes in 1 unit or 5 unit. So if you are using 5 unit of Taq polymerase, then use 0.3 microliter. If you are using 1 microliter of Taq polymerase, then use 2 microliter of it. Then your total amount will be 12 microliter, and you can always prepare it. Just calculate these things before adding your molecular water and genomic DNA. Okay, so firstly prepare your master mix of 12 microliter with these components. Then you can just separate in different PCR tubes, and then add your genomic DNA. Okay, and adjust the amount or make the final volume of 25 microliter with molecular water, because generally we have this amount of 30 to 50 nanogram of genomic DNA we need in our PCR. But generally what we get in after DNA extraction, that we have 3 to 5 nanogram per microliter. Then you will have to add this much of nanogram genomic DNA by adding 6 or 7 or up to 10 microliter of your extracted DNA. Then you can just adjust with it using molecular water and make the final volume of 25 microliter. Then after preparation of this reaction mixture, go for PCR. Using the PCR, you are like you use your smartphone, PCRs also have a different type of screen or features, or you will have to set these things. There are 3 steps. Okay, the final primary denaturation, this is also variable to 2 to 4 minutes. Step 2 is initial denaturation of 15 to 30 seconds, or sometimes 1 minute. This is annealing temperature, where your primer is going to anneal on the denatured DNA. This is variable according to your primer. Like for RBCL, we generally get results on 51.9. On ITS, we generally get results on 55 degrees centigrade, or sometimes we get amplification on various temperatures. And the third here in this step 2, the third one is the extension, where it is generally we set 1 minute for if our PCR product is 1 kilobase pair, or if you are your PCR product will be 1.5 or 2 kilobase pair, then you can just extend this time. Okay, in the step 3, we generally, this is the finalization of 72 degrees centigrade. This is generally done on 5 or 10 minutes. Here your PCR program completes, and the final you can just adjust it to 4 degrees centigrade for infinite time. At this step, you just remove your PCR tubes from the PCR. Your amplification is done. After that, go for Agarose gel electrophoresis. These are the sample wells. I know that you all are familiar with these basic things. These are wells. Just put your amplified products of 3 or 4 microliter with your loading dye, mix with loading dye, and fill the sample. Start the power supply, and we will get the bands. If you are a lucky one, then you will get the bands, and that assures that you have amplified your target gene. And after if you are sure that these bands you get, that this is a surety that you have amplified, and just you can go for sequencing. You can during your gel electrophoresis, you can also use the DNA ladder, which will show that what what is the size of your amplified product. Like here 100 base pair, here 200 base pair. If you are getting bands here, that means your amplified product is of 200 base pair. And for in the final, you can just send your PCR product 8 to 10 microliter to the sequencer, and they will sequence your these amplified product, and after that you will be getting the sequences, the gene you have amplified in your PCR. You will get the sequences after the sequencing, and you can use it for constructing of phylogenetic tree, or you can just using the databases like BOLD system or NCBI, you can just identify your organism. Okay, if you are not familiar that what organism is that. Okay, thank you so much for joining this presentation.